Thanks so much. Great to be here. Give me one moment. I'm just getting the right desktop to share. That one. Okay. Okay, can everybody hear me okay? I'm
Can
assuming
hear
that.
you. It,
Great.
it looks like PowerPoint is in. There we go. Perfect.
All good?
Yep.
Great. Yeah, so thanks very much to Omar for having me here, and thanks to all the other speakers. I apologize if there's any overlap here with the prior talks, but it's always good to to re-emphasize some key points, and definitely want to echo the point that Justin and I'm sure a lot of other speakers have made, which is to always trust your eyes and look at everything. And that really ties in with validation. So today I want to go through some practical validation things and particularly highlight some points in the workflow where things can go wrong. So to start off with, what are we talking about when we talk about validation? So really, validation is verification, right? So what are we verifying? And that can come down to a number of different things, right? At different points in the workflow. So we can be talking about, are these particles my protein? So in picking and 2D classification, is my 3D reconstruction correct, right? So the overall shape, the symmetry, details like ligands. What species are present in my reconstruction? So what's the composition of the entity that I'm looking at? How detailed is the reconstruction? So that corresponds to estimating resolution. And how does that detail vary with direction and location? So that ties into anisotropy, preferred orientation, and local resolution. And then finally, is the reconstruction that you've got the best representation, the most informative representation of the underlying data? And that's where like sharpening and post-processing come into the mix. Okay, so just to like give some brief, high-level principles to keep in mind, and these apply to everything really, to everything that you're going to do in science, but here we're talking specifically about image processing. So number one, and probably the most important one, is always be skeptical of your own data. So always consider the null hypothesis, right? Always consider the idea that the particles you're looking at are not protein, not your protein, the ligand isn't bound, etc. Always approach everything with the utmost degree of pessimism and assume that everything is terrible, and then try to prove that it isn't. Secondly, test and test again, and try to test in, orthogonal ways. So, do different reconstruction and refinement algorithms give the same results? Can you confirm the presence of your ligands or binding partners using difference maps, labels, mass spec, and so on? And finally, the third point I want to make here is really always trust your eyes over any validation metric that you're using. Validation metrics are great. They're super useful to get a global idea of what's going on and to identify hotspots where there might be problems. But all of these metrics can be gamed either deliberately or accidentally. And so you really want to be just looking at the map and your model, right? So for example, if a nominally 3-angstrom reconstruction in CryoSpark or Reliant has no side chains visible, then something has gone terribly wrong, right? Because the FSC, as been mentioned, is just a measure of self-consistency, right? So you want to retrace your steps and see, okay, did something go wrong in post-processing? Is the mask I was using for FSC estimation incorrect? What happened with the initial model, the application of symmetry? Where did things go wrong, right? And so on that point, I want you to remember that a lot of metrics that we're going to use and talk about, which are very useful, only measure self-consistency. They're not telling you anything, magical about accuracy or reality of your structure. They're just telling you that 2 halves of the data are consistent up to a certain resolution. And so there's no absolute substitute as yet for human judgment and careful manual attention to detail. Okay, so going through this sort of step by step, thinking first about validation as it applies to particles, but specifically to picking and 2D classification. The first point I want to emphasize, if you can't, and this one should be like really, ingrained. If you can't see particles, they are not there, right? So if you get a micrograph where you just can't see anything, it does not mean your protein is too small. It means the protein is either not there, or if your particles are smaller, the particles you see are smaller than you expect, you should consider the possibility that your sample is dissociated or aggregated. This happens a lot, right? Something I would highly suggest just to sort of train your brain and to train your eyes on this topic is I would really suggest that you pick a few micrographs manually, right? This can be helpful to train a neural network for picking, like to train a topaz network, for example. But it's also really helpful just to train your own neural network to get a sense of what does my data look like, so that later you can recognize when you're collecting more data, is this good? Is this bad? What does it look like, right? Um, uh, uh, and if you're going to do template picking, I would strongly recommend only using templates that are derived from the same data set, right? And always filter to very low resolution, to a resolution that's much lower than the resolution you're then going to be interpreting, right? So, usually something like 20 angstroms or lower would be appropriate there. And the reason for that last point is the issue of template bias, right? Or as it's often called, the Einstein from noise effect, right? Because what you're doing when you're template picking is you're basically translating a set of references, rotated or otherwise, across the micrograph, and you're looking for the regions that correlate best with the reference. But that means that if you do this on a pure noise micrograph, there will by accident be certain regions that just happen to correlate better with that reference, right? And so then if you average those together, you're going to end up with a ghost of the template, even if there was nothing there to begin with. I will say that, if you're sensible, right? If you actually have particles there, and if you choose appropriate thresholds after template picking, for the picks that you choose to take to actually extract and analyze, real features will almost always dominate, except in cases of extreme overpicking. But just to be on the safe side, an easy way to know that you're not going to get caught by something like this is to filter the references you're using for picking to much lower resolution than the features you want to see in your 2D classes. So if you filter your references to 20 angstroms, and then you see clear evidence of helices in your 2D classes, you can be pretty confident that what you're seeing there is real, because that information was not in the references that you're using for picking. Just to walk you through, the classic example of, template bias, the Einstein from noise effect. This arose from studies of the GP120 trimer, where a structure was obtained to a nominal resolution of, I think, 6 angstroms. But it was obtained using references that were filtered to approximately that resolution. And so that triggered a discussion in the community. And one of the experiments that was done was to take these references, so 3 that are calculated from a PDB and 1 that's a projection of Einstein's face, and then use those to extract particles from complete noise micrographs. And so you can see when you look at these so-called particles, there is nothing there, right? It's all, randomly distributed, or rather non-randomly, in this case, distributed noise, right? If you then go into 2D classification, what you will see is that you recapitulate the features of the templates, even though there was nothing there in the data. And so this is just something to keep in mind, keep in the back of your head when you're processing data, that you can get, ghosts of the references appearing. And so, you should be cognizant of that. And so a similar thing happens in real data and has been well characterized. Usually what you will see, so, if you've got references like this and then you repick, you will see, okay, these ones here, right? These ones here have clear features that were not in the reference, right? That are higher resolution than what was in the reference. Whereas these guys over here, these are Einstein from noise references, right? They've been identified by correlation, but they're not real. And if you look at the individual particles corresponding to one of those classes, you can see that these guys are just junk, right? Whereas if you look at a real class, granted the signal to noise is not great, but you can clearly see, in this case, particles of galactosidase, right, that have both the orientation and the general molecular envelope that we expect based on that 2D class. Okay. Also on the picking and validation side of things, I would just highlight that you should always keep an eye out for unexpected guests, right? So this was from a data set we collected on apoferritin, and we saw all these normal looking apoferritin particles. What's kind of interesting here is we see kind of a gradient where we see normal apoferritin particles in the slightly thicker ice. We see something else in the slightly thinner ice. And then we see what look like kind of apoferritin particles again in the thinner ice, which is a little bit weird, right? So, what's going on here? So when you do 2D classification, you can see, okay, we've got some normal apoferritin classes. We've got some little seeds of iron growing there. And then we've got, some junk classes. And then we have this other one, right, which looks vaguely like apoferritin, but in terms of shape and size, but the symmetry doesn't match and everything looks a little bit weird. If you take that and you do a little bit of repicking and train a topaz model to pick just those particles in this range, you can actually get a reconstruction and identify this protein as peroxyredoxin, decameric protein that came down in the preparation of the apoferritin sample. So always keep an eye out for things that you don't expect to be there. And don't always assume that what you're looking at is the protein you were planning to look at, because it can definitely happen that you get something else coming along for a ride, or in certain cases that something else is the only thing that came along for a ride, and you've solved the structure of a contaminant. Can happen, so always consider the possibility. All right. So just to sort of set the stage, the general refinement scheme in cryoEM will start with a model of some kind, which can be either derived mostly these days from de novo approaches, so from ab initio reconstruction involving stochastic gradient descent as implemented in either CryoSpark or Reliant. And then we'll go through a iterative procedure of projection matching. So matching broadly speaking, projections of the model with matching projections of with your particles, matching your particles with projections of the model, which generates a better reconstruction, which allows more accurate assignment of orientations and so on and so forth. And so, this could come from a related structure, but mostly these days it will come from a de novo approach using stochastic gradient descent. So just thinking about compositional heterogeneity and how to validate the presence of additional subunits or additional binding partners. One of the things I want to point to here is that difference maps, real space difference maps, can be super useful even at very low resolution. So over here you can see an example from the early days of ryanodine receptor structural biology. Where there was a data set with calmodulin, indicated by that plus sign, and a data set without calmodulin. This was at 37 angstroms nominally, with a very severe preferred orientation. But if you subtract these two maps, you get a difference map out, and that allows you to assign the positions of the binding partner FKBP12 and the binding partner calmodulin, and those actually check out, right? We now have structures of RYR1 and RYR2 in the low 2 angstrom range, including in complex with calmodulin and FKBP, and those initial positions that were identified by difference maps at very, very low resolution are absolutely correct. So, that's just to show you that, this can be a robust way of identifying the positions of binding partners. And this can also inform model building, right? When you're building an atomic model, and I'm not primarily talking about model building here, but one of the things that I'd always say is that, when you're building an atomic model, you want to use any and all prior knowledge that you have available, right? And so when we were building some of the initial structures of the ryanodine receptor at very low resolution, so at close to 5 angstroms resolution, just identifying what were the subunit boundaries of this tetrameric protein was very challenging. And one of the things that actually helped us was in an initial processing of the data set, we identified one class that actually had a missing subunit. And by subtracting that missing subunit, from the tetramer, we were able to get a molecular envelope for a single subunit that helped us assign the subunit boundaries of the tetramer. And so, in this case, this was just serendipitous, but in other cases, cross-linking data or negative stain data on subcomplexes or FAB complexes where the epitope can be helpful for the same purpose. Okay. And in a similar manner at higher resolution, we can use locally aligned difference maps between hollow data sets where we have a ligand bound and apo data sets where we don't, in order to locate ligand. So in this case, still on the ryanodine receptor, we have these activating ligands, ATP, calcium, and caffeine. And we had blobs in the density, in the direct density map that we thought corresponded to these ligands. But in order to confirm that, we calculated what we call a locally aligned difference map between the hollow and the apo structures in order to confirm that we were looking at what we thought we were looking at. When I say a locally aligned difference map, what am I talking about? So when ligands bind to a protein, they can modify the conformation. In this case, when ATP binds to the C-terminal domain of the RYR, that whole domain reorients. So if you just calculate a direct difference map, what you end up with is a mess because you are essentially combining the differences that you see due to ligand binding with those that you see due to domain reorientation. So what you can do in order to get a clean difference map is extract out the density around that domain from both maps, so from the hollow and the apo, align it together in real space, and then calculate a difference map. And then you'll get a difference map that corresponds to the difference density just for the local changes, abstracting the rigid reorientation of the domain. So here we've done that for a data set where we had 3 ligands binding, so one where there was 3 and one where there was none. But in order to differentiate between caffeine and ATP and calcium, we also did the same thing for individual ligands. So if you calculate a difference map between a data set where you just have calcium bound and one where you have nothing else, you see only local difference density on the calcium. And if you do similarly between one where you have ATP and caffeine and one where you have nothing, you see no density on the calcium, but you see clear difference density on the ATP and caffeine. Okay, so here is where I want to add a cautionary note regarding ligands. Don't believe everything you read in the paper, specifically in the Nature, Science, or Cell paper. So here we can see an anonymous structure of a GPCR that nominally has a ligand bound to the orthosteric site. We can see what looks like quite nice density for the ligand. If I saw this, I'd be like, oh yeah, there's clearly density for the ligand. There's a butt coming, right? So what happens if we have a look at the raw map? Right. All right. Let's have a look at that. Okay, this is what we see in the raw map. Okay, so the ligand is completely invisible at any kind of reasonable contour. And the protein density is anisotropic and broken up, right? So, okay, to start with, this isn't great, right? This shouldn't be happening. But the other question is, how was the nice density figure that we saw back here, right? How did they make this, right? I can tell you how they made this because there's only really one way to make a figure like that. And the way they made it was by carving the density map around the ligand, which you can do using either PyMol or Chimera/Chimera X. Please do not do this. It's still quite commonly done. If you use surface zone in Chimera X or PyMol carve in PyMol, what will happen in that case is that you will take a map like this, where there's no single contour that's enclosing your ligand, and you will just carve out the density around the ligand and reclose the contour. So carve through the density and cut out all the density around your protein, but show unrealistic contours around the ligand. So if you want to show density just around your ligand, one, I would suggest what I quite like doing is just making a movie like this, where there's not been any dust removal, there's not been any zoning or anything done. This is just the map with the ligand, warts and all. And because you rock it a little bit, you can appreciate the three-dimensionality of it, and you can see the fit or otherwise, right? But for your supplementary figures and whatnot, you're going to want to cut around density around your ligand. You're going to want to cut around density bound parts of your protein. So if you're going to do that, I would recommend in Chimera, at least. I'm not as familiar with PyMol, but with Chimera or Chimera X, use surface zone. Do not use volume zone. Volume zone will cut out like essentially make a new map in which all of the density values have been taken out. Surface zone will just take the surface patches at the existing contour level. And so what that means is, with surface zone specifically, if you end up cutting through a contour, you will see a hole in the resulting submap that you get out. That's a good thing. I want to see holes, right? Because it indicates that essentially no funny business has been done, right? It means that when you cut through a helix, when you cut through the density corresponding to a helix, you expect to see an open contour at the bottom of the helix. If there isn't one, I get concerned that something has been done mistakenly that makes the map look better than it is, right? Okay, so apologies, that was a little extended. Just as a side note here, if you like these kind of figures of maps and whatnot, I've got a package called Chimera X trimmings that I find quite helpful for using Chimera X specifically with cryoEM data, maps, and models. This just has like all of my preferences and shortcuts and whatnot that I find useful, just to show what these look like. So it just allows you to quickly set some display stuff. Particularly, I have like a coot mode button that I quite like for just going through and going around and inspecting things in maps that sort of mimics the display style of coot. And also the sim clip command allows you to just clip a slab symmetrically around the center of rotation, or there's another one that allows you to just repeatedly, get a spherical zone around the center. There are also like a bunch of other things in there that might be useful. And I have a shortcut in that package for making those kind of rocking movies that I find helpful for assessing map and model fit. Okay. Just on another note on ligands and things, here's another example. This is another example of a ligand in a GPCR. Why is it always GPCRs? Don't ask me. But when we have a look at the map, this is what the map looks like, right? And if this structure was nominally 3 angstroms or something, right? I think it may be even a little better than that. But like you can tell, looking where the ligand is, this is not 3 angstroms, and the ligand is not there for any reasonable assessment of what there means, right? I apologize. There seem to be some glitches with the movie. And so one of the issues there, apart from the display of data, is just that often a global resolution will be reported, but few details will be given about the local fit to map or the local resolution in the region that actually matters. And so quite often this happens for GPCRs because GPCRs will be solved as a complex with the heterotrimeric G protein, right? And the soluble heterotrimeric G protein region will be beautiful, very, very nice. But then the GPCR is sitting on top of it, wiggling around and wobbling around. And unfortunately, by Murphy's law, the place everyone wants to look is at the other side of the transmembrane bundle, and that's the place that has pretty much the worst resolution normally and the worst density unless very careful approaches are taken to local processing. So just something to keep in mind. Okay. One of the other things I want everyone to keep in mind, and which is not, I think, appreciated enough, is that you should always be thinking when you collect a new data set, think about voxel size calibration or pixel size calibration. Because depending on how you collected your data or how a facility collected your data, the nominal pixel or voxel size that you get from the facility, may or may not be super precise, right? So you should always be asking, like, how was the magnification calibrated at the scope that you're going to be using? Because once it is calibrated, it's generally stable between samples and data sets for a while at the same mag. But if it's wrong, it generally won't cause you any issues when you're processing your data. Everything will look fine. You'll still be able to fit a model unless it's super, really, really bad. But what it does is it kind of contaminates the literature. Because, for example, if you have two structures of the ryanodine receptor that you align at, and one has the correct voxel size and one does not, if you just align these naively, you'd go, oh, these two structures have different conformations. Maybe that means something. And when in reality, all you're looking at is an artifact of the magnification difference. So this, I think, is actually going to become a problem, particularly now that we have more and more structures that are at high resolution where you can actually appreciate these kinds of differences. I think this is something we're going to have to think about as a community because there are a lot of cryoEM data sets out there that have already been deposited where the magnification is clearly wrong, right? And it might only be wrong by 1%, maybe 2 or 3%, but that's enough when you compare structures, similar structures broadly across the data bank that, like, I think that is going to introduce some issues. But anyway, that's something maybe as a point for discussion. But on the practical level, in terms of like, how do you actually deal with this? So there are a few different ways you can approach it. The sort of historical way that we've used is to calibrate the voxel size by manually fitting a crystal structure, either of the whole protein or a domain in Chimera, and measuring the correlation to your map. So correlation to your map at a range of nominal voxel sizes. And that's what you can see going on over here. So in red, you can see the original map at a particular voxel size. And then we tested all of these different voxel sizes, rigid fit a crystal structure, in this case of the N-terminal domain of the ryanodine receptor, and then measured the correlation in the resulting map. And what you can see is that the original one is not at the maximum. The maximum is over at 1.38. So it's, couple of percent off, right? And that's the kind of difference that can make. So it's definitely something you want to correct for. And it's also something you want to think about when you're analyzing published structures. If you're comparing structures to your own new structure, you want to maybe check that, right? Like check, does this seem like does this seem like it was calibrated well, right? There's an alternative approach, which can be great if you're using gold grids, called cryoEM, which fits the pixel size based on the position of diffraction peaks from the gold. That's I think, a really useful approach as well. And the other thing I want to highlight, which I don't know whether Pavel mentioned or not, but there's a automated implementation of that manual, correlation-based approach that's now available in daily builds of Phoenix called phoenix.magref, which Pavel implemented, and also Randy Reed implemented something similar in Phaser, which I've found those very robust for calibration of pixel size. So I would strongly consider looking at those. Okay. So just getting into resolution, what are we talking about when we talk about resolution? So, here you've just got a representation in 1D of the Rayleigh criterion. So for separating two adjacent peaks where they're clearly resolved, barely resolved, and then unresolved. In practical terms, what I would like you to take away is that you should get an intuitive sense of what different resolutions correspond to, right? So if you look at something like this, or this, you should have a sense in your head of what resolution those correspond to. And like here, what you're seeing is you're seeing broad domain-based features, but you're not seeing any secondary structural features. So there we're talking 20 angstroms, maybe 15, right? If you look lower down here, you're seeing helices, right? Clear sausage-like helices, but no evidence of helicity, no evidence of like corkscrew-like nature of the backbone. So that's usually in sort of the 7 to 9 angstrom range, 6 to 9 maybe. Once you start seeing bulky side chains and definition of the helix backbone, but no carbonyls, no small side chains, that usually puts you somewhere in the sort of 3.8 to 4.5-ish range. And once you start to see well-defined small side chains and just start to see hints of carbonyl bumps, that puts you in the mid-2s. And then at higher resolution, at around 2.1, 2.2, you should see clearly defined spherical waters, ions, and clearly defined bumps for carbonyls. Okay. Just briefly going over the FSC, I'm going to skip through this very quickly because I assume it's already been covered. So what is the FSC? So we calculate the correlation in Fourier space between two reconstructions, each from one half of the data. And then at each cycle of refinement, the resolution estimated by the FSC is generally used to set the resolution limit for alignment in the subsequent iteration. So this might pose a problem if all your data is refined together, right? So if it's aligned to a map calculated from all of your data, that can lead to overfitting. So the classical solution for this is to split the data set in half and refine each half independently. So this is referred to as the gold standard FSC, which is described and introduced very well by Shaw's back in 2012. So as an example of this, the reported FSC at 4.6 angstroms, whereas the FSC versus ground truth is 8 angstroms. The difference between them corresponds to overfitting. This gives you a sort of overview of how we do it these days with the gold standard FSC. And that significantly mitigates overfitting. So it allows you to remove these artifactual radial streaks that you see in the density map. However, the one thing to point out here is that like other sources of overfitting exist, even in this approach. So even though your half sets are treated independently, they're being refined in the same way, right? So you're using the same parameters for both half sets, and most importantly, you're using the same refinement mask. And so that can introduce correlations between the half sets, even without mixing the particles together. And so is there a solution for dealing with that? Yes, there is. The most common solution for this is called high resolution noise substitution. And so what this involves is basically repeating your reconstruction after and calculating your FSC after randomizing phases in both halves of your data beyond a certain resolution. Let's say the resolution where the FSC overall drops below 0.8 or 0.9, right? And then calculate your FSC again. And so that noise substituted FSC, if you think about it, should drop off to 0 beyond that threshold resolution because you have no real data beyond that resolution. It's all noise. And so if it doesn't, that means overfitting is present, which is coming from the mask, right? Then if you subtract that FSC from your experimental FSC, that gives you what we call the high resolution noise substituted FSC, right? Or the overfitting free FSC, right? Or in cryoSPARC, what's called the corrected FSC. And so, here's an example with minimal overfitting, right? So where the original FSC goes out to this resolution, we've chosen a 10 angstrom cutoff for noise substitution. And now you can see that there's a little tiny bit of correlation beyond the cutoff, but not very much. It basically zeroes out, right? If you use a mask that has a very narrow soft edge, then now what you see is you see substantial correlation that extends well beyond that threshold resolution. And so that corresponds to significant overfitting, which you can see here for different masks. So basically by using softer and softer masks, you can mitigate that overfitting. And so this is why you don't want to be using like a 2-pixel soft edge on the mask that you're using for refinement. You want to use something that is a bit broader, that will not show such severe artifacts from the mask edge. And that's also responsible for that dip that you will often see in a corrected FSC in either CryoSpark or Reliant. Just as another example of like pathological FSCs, if you encounter something like this where the FSC does not return to 0, assuming that you have like data beyond that you have particles that have been appropriately binned. If your FSC does not return to 0, this is almost always caused by the presence of duplicate particles, right? Which can either be caused by combining particles from different picking jobs or by performing global refinement of symmetry expanded particle sets. The good thing is there's a very easy solution to this, which is just to remove your duplicates, but it's something to be aware of. If you see this, you should go back and remove the duplicates and re-refine. Another commonly encountered example, if you see a bouncy FSC like this, where you have a sharp drop followed by a bounce in the FSC, this is almost always caused by one of two things. It's either user input error. You've entered the wrong spherical aberration, the wrong pixel size, or the wrong voltage, right? When you were importing your data. That's probably the most common thing. If it's not that, and all your import parameters were correct, it's almost always caused by the presence of uncorrected beam tilt or other higher order aberrations in your data. The good thing about that is that both of those issues are correctable. If it's user input, you can just go back and fix that. If it's beam tilt, you can generally refine those higher order aberrations. So refine beam tilt, trefoil, tetrafoil, whatever it happens to be. And in this particular case, that gives a substantial improvement where we go from having a kind of lousy map at 3.4 angstroms to a very nice map at 2.2 angstroms. This was a very extreme example where we had, I think, about 5 millirads of beam tilt. But you'll see this as well in less exaggerated forms in data that's not quite as badly collected to begin with. Okay, thinking about how resolution varies spatially and directionally. This is something you really need to think about in cryoEM because often we will have dramatic differences in how well resolved our structure is, both spatially across the difference map, or across the density map, and directionally in terms of preferred orientation and directional mobility of domains and so on. How do we do this conceptually? So there are multiple different approaches to measuring local resolution. Probably the simplest conceptually is blocres and variations of that, which basically involve calculating an FSC within a local box or a local spherical mask that's just like translated across the reconstruction such that the resolution value is assigned in a new local resolution map. The resolution value is assigned at the center of each box or the center of each sphere and then like averaged in a window. Obviously, you need to think about in that approach, you need to think about how small are you going to make your box, right? Because there's a trade-off. If you make your box too big, then you lose the value of doing anything local. If you make your box too small, then your FSCs become too noisy and are not terribly informative with regards to the local resolution. So generally what's done is we use, and the approach described in BLockRes is to use what's called an adaptive window factor, which basically means you take your global resolution, multiply it by some fudge factor, usually around 6, and use that as the box size to choose for local calculation of FSCs. And these local resolution maps can be really helpful in order to identify sites of flexibility or mobility, and also to inform atomic modeling, map post-processing, and also just to inform further processing of your raw data, right? So like, for example, if we looked at this, we might say, this domain over here is not so well resolved compared to the bits in here. Maybe it's mobile. Maybe we can improve that by local refinement, with a symmetry expanded particle set, for example, right? The other thing I would point out here is it's super important to consider the local resolution in the site of biological interest, right? That's much more important than the global nominal resolution, right? So for example, if we reported this reconstruction and said, the global resolution is 3 angstroms, which it probably would be, right? But we're doing a paper on something that binds to this domain, then saying that the resolution is 3 angstroms is not super informative. What you want to say is that the resolution at the site of ligand binding locally within a mask around that domain is whatever it is. In this case, like 6 angstroms or something, right? And, that might well be enough to answer the question that you're asking. If you're asking, does a large protein bind in this region? Okay, 6 angstroms might be fine. If you're talking about, details of binding of a small molecule ligand, not fine, right? So it's all context dependent. Okay. So why does resolution vary spatially? This is something to think about. It can be due to compositional heterogeneity, so like partial occupancy of a subunit, variable presence of detergent in a micelle or nanodisk. Those are things you can address if they're impeding your interpretation using either classification or biochemical alterations to the sample. Conformational heterogeneity can also impact. So for example, interdomain flexibility. So that can be something that's purely like structural and not terribly interesting. So for example, a flexible linker between independent domains, or it could be functionally relevant, for example, different states of an enzyme. If that flexibility is constrained, so it's like a hinge-like motion that's only present on one axis, that could also cause map anisotropy in addition to causing local variation in resolution. It can cause directional variation in resolution. This is something that can be addressed by classification, mask refinement, and biochemical approaches. And the other reason that the other thing to think about, if you see a region where you see low local resolution and you don't expect it, right? So let's say in the center of a highly oligomeric protein where the resolution is all beautiful, density is all beautiful, and then in the middle you see a region where you see well-defined density but much poorer local resolution, you should have a think about, maybe this is pseudosymmetry, or maybe I have a symmetry mismatch here. And so in that case, you can address that using classification without alignments, symmetry relaxation, or refinement with the local symmetry that's appropriate in that region. Okay, so I just wanted to show one of these orientation distribution plots just to give you a sense of what they mean, right? So here it is for the ryanodine receptor. These are the different orientations we have for our protein. The way to interpret this kind of plot is it's basically like a map of the world plot. So it's like a Mercator projection of your protein. So if we look at your side views, in this case, the side views are all along the equator, and the top views are all along the poles of the data set. So that's just to give you a context, right? If we had a data set which just looked like this, right, where we only had particles that were present in this region of the parameter space, that would give us a terrible reconstruction. And that would give us something called preferred orientation. And so what that can lead to is a distorted, stretched anisotropic map, which severely hinders interpretation of the density. You can diagnose this using a 3D equivalent of the Fourier shell correlation, where basically you're calculating equally spaced conical FSCs throughout the data set. And so what you see in that, if you do that, you'll see if you have a preferred orientation, you'll see reduced sphericity of the 3D FSC, and you'll also see a large spread in the directional resolution from those conical FSCs that can originate from the preferred orientation. I would highly recommend when you deposit any map or report any cryoEM structure, you should really be reporting the anisotropy. Don't really care how, but you should be reporting and quantifying the anisotropy of your data, because it really matters for the end user in terms of how that data can be interpreted and how what you can get out of it, right? Because if you have a map like this that's severely stretched, you can only interpret it basically as well as the worst direction in your data, right? Okay, so that's one thing. What I would say is, generally speaking, the answer, if you can't deal with it computationally, is try to correct it experimentally. And so that means either the addition of additives, so usually high CMC detergents below the CMC, so things like CHAPS, fluorinated octyl multicide, things of that nature, or collect another data set with a fixed tilt that refills in some areas of Fourier space that are otherwise not occupied. Okay, so there can actually be two reasons though why you might have preferred orientation. Or rather, not preferred orientation. Let me rephrase. There can be two reasons why you might see an anisotropic density map. One of those reasons is preferred orientation, which we've sort of discussed, where instead of adopting random orientations in the ice, those particles adopt a limited subset of orientations. Often this can be due to association with the air-water interface. It can also be due to the fact that certain orientations are just hard to pick, right? If that's the case, you can address that by careful particle picking, training a neural network. There have been some good examples of that recently. If it's the former, though, if those orientations are actually missing, you can address that using additives, tilting, or sometimes by classification, where you discard the preferred views and isolate a subset that is isotropic. One thing I would say, though, is often if you do have a genuine preferred orientation, because that involves your protein associating with the air-water interface, that's often additionally associated with particle dissociation or damage to your protein. So I would generally favor a biochemical fix to this problem if at all achievable. However, the one other thing that I want to mention is that anisotropy is not always arising from preferred orientation. It can also arise from conformational heterogeneity. If you have two domains which are arranged like this and they're moving like that, so you have a hinge, that will lead to the same thing. That will lead to anisotropy because the density is going to be blurred out in the direction of motion, right? And it's going to be okay or better at least, when you're looking orthogonal to that. So that's something to consider. If in that case, you can potentially correct that by local refinement and classification. Okay, just to give you a concrete example of what I mean. So here's an example from the literature. It's another GPCR, sorry, load of GPCRs. If you look at it in this direction, you might say it looks okay, right? Like you can see, okay, we've got helices that look kind of helical, and in the center there, maybe that's all right, right? Turn it 90 degrees though, and it's a different story, right? Like everything is super, super, super stretched, and interpreting this map is going to be a real challenge, right? But if you just look at the global resolution, this is going to come out at 2 point something, high twos maybe. So if you just report the global resolution and a picture of the map that's in that direction, that's not going to be a very informative for the user who's trying to understand, what's the data? What does the data look like? What's the basis for this atomic model? And so that's why I would strongly, strongly encourage if you're using CryoSpark, there's a comprehensive package of orientation diagnostics that are now calculated on the fly during refinement and can be calculated also afterwards using this orientation diagnostics job. So that is really great for providing metrics for anisotropy in an easy to digest format. The conical FSC area ratio is particularly useful in my experience. And you can report all this data directly. That's really nice. Similar data is reported using the 3D FSC server. Just pick one. One is better than none. Just show something that shows how anisotropic your data is to inform the users. I actually favor really basically these plots which show the spread of directional resolution. I think they're really nice. And the histogram, the overall histogram showing the directional resolution of the conical FSCs, I think is really nice because that gives an intuitive answer for like how the direction varies. And generally, I find that histogram correlates pretty well with what I see visually. Okay, so just showing an example of mitigating map anisotropy by master refinement. So here, this was a Class C GPCR I was working on with Ching Fan's lab. You can see in the before image, it's very anisotropic, difficult to interpret, and it's stretched along one direction. If you perform careful master refinement of this region of the protein, you can really substantially mitigate that issue. And so this is an example where anisotropy is due not to preferred orientation, but primarily due to interdomain flexibility. I would say that, if all else fails, you can use some of these machine learning-based map improvement algorithms like Deep Enhancer to improve map interpretability. However, as has been noted, caveats abound. And particularly, I would also note with regards to ligands, they can just disappear. So if you're going to use something like Deep Enhancer, and it can be useful, like we've used it, it's been useful, but always make sure that what you're seeing is present in the ground truth, right? Always look at it in conjunction with the original map and only interpret features that are present in the original map, and be aware that some features may disappear from the original map when you process it with Deep Enhancer or EM Ready. I would also point out there's a really nice new approach from Yifan Cheng's lab called AR Decon, which is a non-machine learning based approach to map deconvolution. And that can also give, can rescue some maps that are on the edge of being interpretable. But ultimately, I would always tend to say you're better to go back to the grid in cases like this if you have the capacity to do so. You can also mitigate map anisotropy by classification. So in this case, in the ankyrin complex, we have these very flexible transmembrane binding partners that are linked only via interactions of the cytoplasmic domain to ankyrin. And so if you just do local refinement of one of these domains, or one of these proteins, band 3, after global refinement, you get a pretty lousy map at about 4.5 angstroms. It's better than what we had initially, but I wouldn't want to build into this. If you do 3D variability analysis, you can actually identify a component that corresponds more or less to an order-disorder transition. I call it an order-disorder transition. It's probably not physically that, but it allows us to identify clusters of particles that are better ordered. And if we take those clusters and then redo global refinement for the whole thing, redo local refinement, we can get a substantial improvement in the local map that's much more interpretable. You can see bound lipids, you can see our single transmembrane helices of glycophorin are a lot better resolved, and so on and so forth. And if you do that overall, for different subunits of the same complex, you can obtain a composite map that allows us to interpret the relative orientations of the different binding partners in the complex. Keeping in mind, by the way, that like this presents a kind of misleading picture of what's going on in reality, right? Because if you just looked at this de novo as a composite map and treated it like a non-composite map, you would say, oh, this whole thing is super rigid, right? Everything is really well defined, which is not the case at all, right? All of these band 3 molecules are moving around like crazy, right? So what's the best way to deal with this kind of data? I don't know. I think the best that we've come up with is we deposit a composite map, we deposit the original map, we deposit all the local refinements and the corresponding atomic models, and we describe it in the paper. I don't know that there's a better way to do that, but I think there's definitely room for improvement there. Okay. Whoops, sorry for that. Okay, so that's about it. Just general principles come to be in mind, coming back to them to re-emphasize. Always be skeptical of your own data. Always consider the null hypothesis. Test, test, and test again, and trust your eyes. If your 3-angstrom reconstruction has no side chains, have another look at it, right? If you can't see your ligand, even though you think it should be there, consider the possibility that it's just not there, right? Also want to highlight, this paper on community recommendations for cryoEM, archiving and validation, some really good recent recommendations for the community and well worth reading and discussing that, I think. That's all I got. Thank you all for listening, and I'm happy to take any questions.
All right, thank you so much for a really thorough, really wide scope presentation there. We've got a number of questions. You spoke about the value of using difference maps. Can you describe your procedure for generating those difference maps?
Yeah, sure. So specifically what I was talking about there was locally aligned difference maps. I was doing that in Chimera. You can also do it in Chimera X. So what I have a shortcut for this, I think in Chimera Chimera trimmings or Chimera X trimmings. I have both. But basically what I do is fit a model to one of you've got your both your maps globally aligned together. Fit a model to one of the maps, zone out a volume generously around the domain that you think your ligands are binding to, right? And then align both of your maps together. So just use fit map to align both of your maps together and then subtract them. And that will generate a difference map. And then you can also use a little trick to put that difference map back in the right frame of reference for either the APO or the holo maps, depending on what you want to do. And then, yeah, that's basically it.
So there was a question about how one calibrates their pixel size. I wanted to comment a little bit about how, as a center, we handle this because we report these values to our users. We routinely will collect apoferritin data, come up with a reconstruction, scale it to Shores' high-res map, and then apply that scaling factor to their APIX, which we trust. And that's the value we then provide. So if you'd like to add a little bit about how someone who is receiving data, once they've built a model, how can they be confident that they've been working at the right pixel size?
Yeah, I think that's great. I would say that, that's what should happen at every center. It's not what happens at every microscope or every facility. And also you don't necessarily know when the last apoferritin data set was collected, all of those things, right? So with all of these things, I would tend to take an approach of, trust but verify, right? Where just talk to whoever's collecting the data, ask them, when was it calibrated? How was it calibrated? And go from there. I would generally recommend calibrating with a crystal structure rather than another cryo-EM map, just because in the case of a crystal structure, the wavelength of the X-rays you're using and the detector distance are pretty precisely known. For us, what we do is if we have a domain in one of our structures where there is a crystal structure, we will use that for calibration. And typically these days, And by these days, I mean the last couple of months, we'll use phoenix.magref because that is a more robust approach to what we were already doing manually. So that's really nice to have, I think. And yeah, I would generally just think it's something that users should keep in mind, right? Because you're often collecting at multiple different centers, and you should always keep in mind the possibility that something is not quite right, and double check when you receive your data, how was the pixel size calibrated? Because if it was just calibrated using a waffle grid, then it's not going to be accurate enough for what we want to do.
Paul, if you'd like to add something.
Thank you. Yeah. Yes. My colleagues for a couple of years ago published a paper and a Python package. It's called EMDA, EMDA, Electron Microscopy Data Analysis. And it is a toolkit for, well, as you say, data analysis, Fourier correlations. And one of the things that it can do, section 4.2.2, I think, in the paper, is about magnification refinement.
Thank you. All right. For decades, resolution has been the first parameter of choice for crystallographers, but it seems that in cryo-EM, it's a little more difficult to pin down an actual resolution. Do you have a suggestion for what metric should be used as a quality cutoff?
I would say you know it when you see it, which is not a terribly useful answer. But I think what it comes down to is people should be showing their work, right? If you report a 3 angstrom structure and it's obviously not 3 angstroms in the regions that you care about, that's going to be abundantly obvious, and is abundantly obvious. So in a sense, we can talk all days about different metrics used for this kind of stuff, but what I would say is just show the actual density, right? If you see a map like the one I've got in the background, right? There's no argument, right? The data should speak for itself. If it doesn't, if you have to come up with fancy statistical, metrics for whether or not it's appropriate, then generally speaking, it's not. Yeah.
While we're on the topic of resolution, say you've got a situation where a region of interest has a local resolution of 6 angstroms, and local refinement yields a resolution that's substantially different, say 3 angstroms. How does one actually describe the resolution of that region? What number should one be providing?
Well, I would just say the resolution after local refinement in the region of interest was 3 angstroms, right? Because you're going to deposit that as a separate map anyway, right? You're going to deposit that local region. Let's say it's around the ligand binding site, and assuming that all the features of that map check out, and obviously with local refinements, you also got to be careful because you're using a smaller mask. Usually, overfitting can become more of a concern, so you want to be careful that you're using a mask that is soft enough, right? When you're doing local refinement. But yeah, if you see substantial improvements in density that correlate to that estimated resolution improvement, report it as is, right?
You described an example where data was collected with a significant amount of beam tilt, and after correction, the resolution was a little bit over 2. Can you remind us of how much beam tilt there was in that data set, and do you have a threshold for what's too much?
It's too much if you can't correct it. So that data set had about 5 millirads of beam tilt. I can't remember whether it was 5 millirads overall or just 5 millirads in the worst image shift groups. But either way, it was pretty extreme. And that data is on Empire, actually. We often deposit our data on Empire, and I want to give a big shout out to Empire because it's been super helpful for us in developing processing workflows and so on. And also, it's a fantastic free data backup service for our for difficult data. But seriously, it's been super helpful. And it gives us a lot of examples for the community where there are things there that, you know, we can process that with new approaches can go to much better resolution than they could before, and it allows us to improve our workflows. And yes, so that data that I referred to, that was actually that's actually a neat story. It was one of Francesca Valese's projects while she was in my lab, where we accidentally purified a reaction center from E. coli, the cytochrome B03 oxidase, and reconstituted it in nanodisk, and didn't realize until we collected 1 day of Krios time that it was the wrong protein. But because it was a membrane protein in a nanodisk, right? It looked kind of like what we expected to until we looked at the details. But it went to 2 angstroms, and so that did end up getting published, and the data is on Empire for anyone who wants to look at it.
So a couple of questions about preferred orientation. You described using an approach called masked refinement. Could you go into a little bit of detail about what that is?
Oh, so that's just local refinement, right? So instead of refining with a global mask around your entire protein, this would be in a case where you have 2 domains that are essentially hinging with one another, right? And so in that case, it won't deal with preferred orientation, but it can deal with map anisotropy due to interdomain flexibility or interdomain mobility. And so then what you would be doing is starting from your initial particle orientations from the global refinement and performing a local search of particle orientations with a mask just covering 1 domain.
In instances where particles have very different dimensions along different axes, you can imagine introducing a preferred orientation issue by just picking with a diameter that might be too small or too large. What are your suggestions for, 1, being able to recognize that, and 2, addressing it?
Yeah, it's definitely something that can happen when you have anisotropic particles and you look down the axis, they can just look like dots, right? Quite common for RNA things and different things that like have that kind of nature. One thing that we've found really useful lately is using micrograph denoising. I quite like most of these approaches use the noise to noise algorithm, but they do it in different ways. The recent implementation in CryoSpark works really well. And so our sort of default workflow these days would be, you know, patch motion, patch CTF, micrograph denoiser, and then blob pick, and also manually interpret the micrographs from there. And the denoising really helps with manual interpretation, particularly for small particles or for particles that have small cross sections like you're describing.
Is it possible to generate difference maps in CryoSpark?
Don't think so. There's no Boolean,
Okay.
there's no map subtraction capabilities in CryoSpark. We do all that in Chimera or Chimera X. You can also do it in Phoenix. Phoenix has a tool for everything, and I'm pretty sure there's a tool for making difference maps.
Jumping back to the pixel size issue, if one refines a model against a map whose pixel size is incorrect, are the atomic coordinates different from where they would be were the map at the right pixel size? And can one observe stereochemical strain?
Yeah, it would be nice. Unfortunately, usually, so the model will definitely be globally different. It'll be shrunk or expanded, right? But the problem is often you're looking at a shrink or an expansion of somewhere on the order of, you know, 0.5% to 2%, right? Which is small and you're not going to see that, generally speaking, unless you're at ultra high resolution. That's just going to be taken up by whatever fudge factors you have for refining bond lengths, bond angles. You know, it's unfortunately not going to show up clearly in the data.
There's a comment about calibrating pixel sizes based on diffraction off a cross grading, and that this person always gets pixel sizes that are about 3 to 5% larger than those calibrated by using a crystallographic model. I can speak a little bit to this in that I know some of the cross gratings have multiple lattices, and that first ring is actually 3 or 4 rings all merged together. And so it can be difficult. We actually have tried Chris Russo's MagCal approach, and that agrees within 0.5% of scaling to APO maps that I described earlier. But this person is wondering if that's something that other people notice.
Yeah, I mean, we don't rely on any calibrations that are done with cross gratings. We would always calibrate with a crystal structure.
All right. For flexible particles, which we expect to see more and more of, should we instead be depositing movies instead of single coordinate sets?
I don't know how you would do that, to be honest. I think, you know, there's a case probably for depositing, like if you're doing 3D VA, depositing sets of maps that correspond to, you know, traversing one particular coordinate, right? I think there's certainly a case for depositing maps that are like reconstructed along a 3D VA coordinate, if that's important to like the biological question that you're making. And then people can make a movie from that if they want, but it still retains the 3-dimensionality of, you know, of a proper map. How exactly you deposit that data is not totally clear to me, right? You don't want to have 50 additional maps in your EMDB entry, right? So right now, when people have done that, they've generally done it via Zenodo, but some way to more officially support that by the EMDB, I think would be helpful.
Bit of a philosophical question along these lines. Given that each protomer of a complex would have its own set of dynamics, should we ever be assuming symmetry?
I mean, yes, you should be, because mostly that's not the case. They're the same for the question is, you know, you can also, what do you do if you don't assume symmetry? Because if you just refine in C1, that is not assuming symmetry, because you're still going to be averaging things based on alignment to a symmetric molecule, right? So in a perfect world where you had, you know, data to such a resolution that you could align everything to 2 angstroms and actually tell the difference, fine. But we don't live in that world. So I would use symmetry when it's helpful, but be cognizant of the fact that it's rarely perfect and, you know, take account of that.
Local refinement in cryoSpark sometimes gives varying CFAR values when using different settings. What should be done in cases like this?
So yeah, metrics for anisotropy, I think, is still evolving, and there's still something. And so one thing that I didn't really get into when I was talking about this is there's a question as to, so let's say you have a preferred orientation, right? Meaning that you have some orientations that are missing from the data set. In a perfect world, like if you just take a data set where you have orientations assigned, you take some of those orientations out, take quite a lot of those orientations out, and then just reconstruct a map, the map will look more or less fine, right? It won't look terrible or stretched. And the reason for that is that preferred orientation per se, like in my mind, is not primarily an issue of missing orientations. That's the origin. The problem comes from misalignment, when the things that are in the dominant orientation end up misaligned to the orientation that is missing. So that's one thing to point out. The second thing to say is that we've encountered cases that have, and I've reported this on the cryoSpark forums, we've encountered cases where the CFAR value looks terrible. Like it looks like 0.1 or 0.05 or something, but the map is beautiful and very interpretable. And like if you dig down and have a look at the conical FSCs, what you will see is that there's like there's one region of Fourier space that is missing, genuinely missing, and there's a lousy resolution in that direction. And so if you look at the histogram, you'll see a bunch of directions that are fine, and then one or two directions that are at like 12 angstroms, because whatever reason that orientation is just not there. And so that gives a CFAR value, which is defined by the conical, the area under the conical FSCs at the maximum over the minimum, right? Or the other way around, whatever. Minimum over the maximum. So that'll give a CFAR value, which looks terrible. But if you don't have a misalignment problem, then actually the map looks fine. So I think we don't have the perfect solution for this yet. What I would say again is like, you should just be looking at the maps, right? And it's something, they're useful metrics to guide you and you should report them. But ultimately, if the map looks isotropic, the map looks isotropic. That's ultimately what you care about.
And that is our last question.